Okay, welcome back to Part 1b of Lecture 17. We're going to be talking about body fluids a little bit more, and we're going to go into a little bit more detail about water balance. And we'll come back and revisit the compartments and talk a little bit about how the fluids move between compartments. This material, once again, is from Chapter 26 in Marib. Now let's first talk about body fluid ionic composition. We mentioned this before, but we want to go into a little bit more detail about the ions that are in the different fluids in the body. Let's start by looking at the graph that's on the right side. You'll see that the purple bars represent extracellular fluid. The green bars indicate intracellular type fluid. And at the bottom of this graph, you'll see that we have the different kinds of ions or electrolytes that we have, for example, sodium, potassium, calcium, so on and so forth. Now, one of the things I would like you to know is I would like you to know the chemical symbols for these, and I would also like you to know the valence of each of these ions. So, for example, sodium and potassium, these are monovalent ions, in other words, have a 1 charge, so it's chloride, whereas we see divalent ions like calcium, magnesium, sulfate, okay, and then we have a trivalent ion like phosphate, and bicarbonate here is with one of the important ones. Remember, this is univalent or monovalent as well. Okay. The other thing I want to mention about these ions in the bottom is that some of these are what we refer to as polyatomic ions. In other words, if we look at bicarbonate, one that we're already familiar with, and we look at the chemical composition of this is hydrogen, carbon, and oxygen. This tends to stay together as these 3 different atoms when we put it into solution. So when we put bicarbonate in solution, it doesn't dissociate into hydrogen, into carbon, and into oxygen. It stays together as a bicarbonate ion. Same thing is true here as you see with phosphate and with sulfate as well. So with that little bit of introduction, let's take a look at the graph and take a look at where we find these ions in the body. First of all, we said before that the extracellular fluid ions are represented by the purple bars, and we can see the most significant ions that we have in the extracellular fluid include sodium, a big bar over here, that's going to be chloride, and one over here that's going to be bicarbonate. So as you see on the left side here, the major extracellular fluid ions that we have in the body include sodium, chloride, and bicarbonate. Now if we look at the intracellular fluid ions, we see a big bar here for potassium. We see one over here for magnesium, and we see another one over here for phosphate. And the same thing is said over here. Basically, the major intracellular fluid ions are potassium, magnesium, and phosphate. Realize that inside cells, we also have negatively charged proteins. So these could be considered ions or charged particles, but remember, these are too large to get out of the cell, so these are inside cells. So these somewhat offset the positive charges inside the cell. Now we see 2 ions in the bottom that are pretty much a toss-up as far as being extracellular and intracellular, approximately equal or so concentrations intra and extracellularly, although we do see much more calcium in the extracellular fluid compared to what we see in the intracellular fluid. Okay, so that's an important graph. You should know the things that are on the left here, and as I said, and as it says here, you should know the chemical symbols for these, and you should also know the valences of these different ions. Now let's continue on and talk a little bit about movement of fluids between compartments. We defined the compartments, remember before, as being several in the body. So we had, for example, the intracellular fluid compartment, which is here. Remember, this constitutes about 67% or so, about 2/3 of all the fluid we have in the body. And all the rest of the fluids that we see in the diagram here constitute about 33% or about 1/3 of all the other fluids. As you can see by these arrows, we have movement between the different fluid compartments. Whenever we talk about fluid movement, we really have to come back to those same forces that we've been discussing all throughout A&P. That is hydrostatic pressure and colloid osmotic pressure. These are the ones that will determine whether fluid moves one way or the other. We talked about capillaries in exchange with the interstitial fluid, and we mentioned that it's the hydrostatic pressure that basically forces fluid out of the capillaries, and it's the osmotic pressure due to dissolved solutes inside the blood vessels that pulls the fluid back in. So these 2 forces were at work here in the systemic capillaries to exchange fluid with the surrounding tissues. And remember, when we talked about the kidney, we also talked about the capsular hydrostatic pressure as well. That was another type of hydrostatic pressure we talked about. Now remember, when we talked about the fact that capillaries exchange fluids with the interstitial tissues, that we have about 3 liters or so extra plasma fluid that's deposited in the interstitial tissues that isn't returned to the capillaries, and that, remember, is brought back by the lymph. And that was the purpose, one of the purposes anyway, of the lymphatic vessels. Now we have a couple of others. You'll notice that we have an intracellular fluid exchange with the interstitial fluid as well. So fluids are always moving between cells. And of course the driving force for this really is essentially the hydrostatic and colloid osmotic pressure. Remember that water is going to follow solute, and basically we're talking about the buildup of fluid in one place that causes a hydrostatic pressure that tends to push the fluid somewhere else. And so this is both forces are involved here. The last type of fluid that we want to talk about is what's known as transcellular. We didn't spend too much time on this yet. We want to talk a little bit about this here. This is the type of fluid that we don't characterize as either interstitial, lymph, intracellular, or plasma type fluid. This is a little bit different. We have a continual exchange of fluids across transcellular membranes. And really what we're talking about here is, for example, water movement across mesothelial surfaces, like the peritoneum, like the pleura, like the pericardium. We also have this movement across synovial membranes. Remember, those are in synovial joints. And we also have this between the blood and cerebrospinal fluid, between the fluids in the eye and the ear. So all these different places where we have fluid movements crossing these membranes, we basically refer to this as transcellular fluid, and here you can see the serous membrane that we're talking about. Now once again, just to remind you, the net movements of fluids between compartments then results from differences in both hydrostatic and osmotic pressure. These are the two drivers of fluid movement throughout the body, so that's kind of important for you to remember. Let's turn our attention now to water balance or fluid balance in the body. In this diagram, you'll notice we have two bar charts, and on the left side you'll see it says total intake 2,500 milliliters. On the right bar it says total output 2,500 milliliters. So remember that we're talking about a balance here, and that means the amount in equals the amount out. So typically in a normal day we take in about 2,500 milliliters of fluid, and we excrete or put out about 2,500 milliliters of fluid. So normally we're in balance. This doesn't mean, of course, that we're in balance every single day. This is really over the long run, over the course maybe of a week or 2 weeks or so, that our body normally achieves the balance that we see that's here. So this is not on a moment-to-moment or day-by-day basis. This is really on a kind of longer term. But in essence, our body will always maintain balance as long as we're physiologically healthy. Now let's take a look at the sources of water. That is, let's take a look at where we get our intake of water every day. One of the things you'll see on the top here is what's called the water of metabolism. This simply means the water that's generated during aerobic metabolism in the mitochondria. Remember, the two things that mitochondria generate as a result of making energy was, of course, ATP. The second thing was water. The oxygen, which is the final electron acceptor, merged with hydrogen ions plus electrons that run the electron transport system, and all of those combine to make water, and that is called the water of metabolism. This is about 10% or so of the total intake or total generation of water that we have. The second item that we have here is water in moist food. Now what we're talking about here is not some of my favorite foods like ho-hos, ring-dings, those sort of things. We're talking about things that have lots of water content, for example, fruits, vegetables, and those sort of things. This is about 750 milliliters, or about 30% of our total water intake every day. And finally, you'll see down the bottom, the largest category of intake as far as fluid goes, or water every day, is water in beverages. Whether we drink water or beverages containing water, this constitutes about 1,500 milliliters, or 60% of the water intake that we have every day. Now let's turn our attention to the average daily output of water. These are the ways in which we lose water from the body. You'll notice on the top here we have about 150 milliliters of water lost through sweat every day. And this is basically from an average person. This is not an athlete. This is not somebody who's doing very strenuous work. This is on average about 150 milliliters, or about 6% of that 2,500 milliliters. We also have water that's lost in feces every day. This is about 150 milliliters. And remember, thankfully, that the 1,500 milliliters of fluid that comes from the small intestine into the large intestine, only about 150 milliliters of that is lost as body water in the feces. We also have some water that's lost through the skin and the lungs. Now this is something that we don't see, and for that reason this is sometimes called insensible perspiration or insensible water loss. And we don't see it normally, but under special circumstances we can see it. A good example is when we put our hand on the top of a lab tabletop, for example, that's black, and the tabletops usually are cold. If you leave your hand there for a little while and then take it away, you'll notice the outline of your hand traced in water vapor. And that tells you that there is water vapor seeping out of your skin. It's very minimal, but it does add up, and it constitutes about 700 milliliters per day. The other source of this is through the lungs. We normally don't see the water vapor that we exhale, except if it's a very cold day. And when it's a very cold day, what happens is the warm air from our mouth or nose basically is exposed to the cold. It condenses very quickly and forms the water vapor that we see. So those two things tell us that there is water loss through the skin, through the lungs. We just don't see it most of the time. And for that reason, we refer to this as insensible type water loss. And by far the largest loss of water that we have is through the urine. We used an average of about 1.8 liters, if you remember before when we talked about the urinary system. So this is right just about on the money here. So we lose about 1,500 milliliters or so, or about 60% of that 2,500 milliliters every day through the urine. So the question then becomes, how do we regulate all this? How does the body regulate this so that the total intake is going to equal the total output? Well, by far the primary regulator of all of this is usually the urine. So the urine production is really the most important regulator of water balance and how we determine that we make the water in equal to the water out. And we're going to see a couple of other ways as well in the next slide. Okay, so let's talk a little bit about water balance and extracellular fluid osmolarity. First of all, let's talk about water intake. What regulates our water intake? Well, the main thing that does this is what's called the hypothalamic thirst center. And in turn, that's stimulated by the osmotic pressure or the osmolarity of the extracellular fluid. When the osmolarity rises, in other words, when body fluids become more concentrated, what happens is there are neurons that are stimulated in the hypothalamus that actually stimulate thirst and stimulate us to drink water. And that allows intake of water to replace the water that we're losing every day. Now, how do we regulate water output? Well, remember, first of all, we have these things that happen, as we talked about in the last slide. Those water losses that we have every day, including the insensible water losses, the water lost in feces, and the water lost in urine, we refer to these as obligatory water losses. And the reason for that is these must happen. We can't do anything about these. We always lose the water from the lungs, the skin. We don't slow that down. We always lose water in feces, as long as, of course, we're eating a normal diet. And we always lose water in urine. In fact, we have got to lose about 500 milliliters of urine a day and put that amount out in order just to get rid of the solutes that we need to get rid of every day. So this is really a minimum of urine that we have to put out every day in order to remain healthy or in homeostasis. And so all these things add up, and this is the what we call the obligatory water losses from the body. Now, what happens is when the extracellular fluid becomes more concentrated, we set up here, one of the things that happens is we stimulate thirst. The second thing that happens is when those hypothalamic osmoreceptors detect increased concentrations of body fluids, they also stimulate the release of ADH via the posterior pituitary. And remember, ADH is basically a water conservation measure. Okay, so this doesn't take new water into the body. This basically helps us to conserve the water that's already there. So antidiuretic hormone acts, remember, at the collecting tubules or collecting duct to bring back some water into the body. So we retain water when ADH is secreted. And just to mention this, when we have any large changes in blood volume or blood pressure, remember, not only is ADH released, we would also have renin released to basically reabsorb salt and water as well and to restore normal blood pressure. Okay? All right, so let's talk a little bit about fluid imbalances now that we've talked about how the body maintains balance of fluids. And you'll notice that there really are two major forms of fluid imbalance that we can have. We can either have a fluid deficiency or we can have a fluid excess. And you'll notice that in each of these categories we can have two major types of these. Okay, so let's talk a little bit about this. First, let's start with fluid deficiency. Let's talk first about volume depletion. And we call this hypovolemia. This is a good term for you to know. What I want you to notice is that when we talk about volume depletion, we're saying that the total body water is reduced, and this is an important point. The fluids that we're losing from the body are lost in an isotonic fashion. What does that mean? Well, think of hemorrhaging, for example, if we're losing blood and blood plasma. That plasma is isotonic to body fluids. And if we're losing a lot of that, we're losing the same proportions of salt and water that we normally have in the body. That's why we say that this is an isotonic type of loss. This is going to deplete volume, but it is not going to change the osmolarity of body fluids since we're losing an isotonic type fluid. On the other hand, let's take a look at dehydration. The word dehydration, of course, means to take water away. What we're doing here is we're going into negative water balance. In other words, we're losing more water than we're taking in every day. We're not talking about salt here, we're just talking about water. Once again, this is going to reduce the total volume in the body. However, what we're going to do now is leave behind hypertonic body fluids. Why? Because we're taking water away from the normal body fluids and we're leaving behind the salts. So dehydration is water loss only, and that means that we're going to leave behind a hypertonic body fluid, so that the osmolarity of body fluids is now elevated after this water loss. Let's turn our attention now to fluid excess, and once again there are two types of this. We can have a volume excess, and what we're talking about here is elevating the level of fluids in the body, but doing it in an isotonic manner again. So here we're putting into the body isotonic fluids, and all we're doing is expanding the volume of fluids in the body, but we are not changing the osmolarity. On the other hand, let's look at hypotonic hydration. What we're talking about now is taking in basically pure water or anything that is less osmolarity than normal body fluids. We would refer to this as hypotonic hydration. And we're going into positive water balance in here. This can ultimately result in something that's called water intoxication, which we're going to talk about in the next slide. Notice that we're still elevating the total amount of body fluids, so we have an elevated body water content. However, what we're doing now is introducing hypotonic fluids into the body. What this is going to do is lower the osmotic concentration of all body fluids, and so we'd say that now the fluids in the body are hypotonic to what they should be, because all we've done is just add more water without the salt. So this is a good chart for you to come back to and kind of think through a little bit. This will give you a little bit better idea about how we change the tonicity of body fluids. Okay, so let's talk in some detail now about these two situations. We want to talk about either dehydration or overhydration, which we kind of mentioned on the last slide. Remember, these are examples of removing or gaining only water. So we're only talking about changing water content, not salt content. So let's talk about dehydration first. If we take water away from the body, that means we're leaving behind salts, and those salts are going to be increasingly concentrated, and so that the osmotic pressure increases in the intracellular fluids. And as we talked about, what will happen is we'll stimulate the thirst center to stimulate drinking, and we'll also stimulate the posterior pituitary to secrete ADH. So ADH is going to be water conservation measure. Stimulation of drinking through the thirst center is going to bring new water into the body. Now, what I want to talk about in a little more detail is this right here. The fact that when we have a dehydration in the body, water moves out of cells. Okay, so the cells ultimately shrink. What I want to do is take a closer look at exactly how that happens. So let's begin with this down here. This is a blood vessel. And in the blood vessel, of course, we have an osmolarity of about 300 milliosmoles per liter, right? Normal body fluid osmolarity. And we're going to imagine that up here we also have a body cell. And of course, in between we would have interstitial fluid. Now, one thing to realize is that the interstitial fluid is isotonic to the plasma. So this is also 300 milliosmoles per liter. And the cells are also isotonic to the interstitial fluid and the plasma. So everything is stabilized here at 300 milliosmoles per liter. As we said, this is about normal body fluid osmolarity. Now we're talking about dehydration. So let's imagine that we're going to move water out of the plasma. Something is going to happen where we're going to take pretty much just water out of the plasma. Sweat, in case you didn't know this, is a hypotonic type fluid to body fluids. So in other words, when we sweat, we lose primarily water versus salt. Let's say that we're taking the water out of the body, and when we do that, what's going to happen to the osmolarity? Well, if you remember our equation, we said that the osmoles per liter is equal to the solute concentration divided by the volume of water. And we said that if water decreases because it's inversely related to the osmolarity concentration or the osmolarity of body fluids, this would increase. So by taking water away, what's going to happen is we're going to increase the osmolarity. Let's say it goes up to 300 milliosmoles per liter because we've taken water out. Now the important thing to notice here is how do we compare, or how does the interstitial fluid osmolarity compare with that in the plasma now? Right, the interstitial fluid is now hypotonic to the plasma. And that means that the interstitial fluid has more water than the plasma does. And so what's going to happen is water is going to be pulled from here into the plasma. And that may adjust this a little bit. This may bring this down to about 300 milliosmoles per liter. So it's bringing it back down toward normal body osmolarity. But when we take water away from this compartment, notice that we're raising this to, let's say, 300 milliosmoles or 310 milliosmoles per liter. So we've actually increased the concentration of interstitial fluids because we're losing water from that. Now, compare once again the osmolarity of cells with the osmolarity of the interstitial fluid. We notice that the fluid inside cells is hypotonic to the fluid now in the interstitial fluid, and that means that we're actually going to pull water out of the cells, and that's going to increase the osmolarity inside the cells. So it's going to make the intracellular fluid hypertonic to normal body fluids as well. So this is really kind of like the first round of adjustments that happens. Ultimately, what's going to happen is the osmolarity here, the osmolarity in the interstitial fluid, and the osmolarity in the plasma are all going to even out. But because we've lost water in the body and haven't replaced that yet, all of these osmolarities are going to wind up being higher than normal. In other words, the body fluids that are left behind are going to be hypertonic. And let's just say, for example, that they all even out at about 307 milliosmoles per liter. That's going to be the concentration of the plasma, the interstitial fluid, and the intracellular fluid. So you'll notice that by taking water away from the body, we've increased the osmolarity in body fluids to make it hypertonic. And this hypertonic fluid is going to now stimulate the hypothalamus to make us drink water, and it's also going to stimulate the hypothalamus to secrete the ADH from the posterior pituitary. Now, what's the effect on cells? Well, it's kind of like a plant. If you haven't watered a plant for a while, what's going to happen to these cells when we take away water, which we did here, is that these cells are basically going to shrivel up and shrink. Hope you remember what we call that from A&P1. That is known as crenation. And we talked about that in connection with red blood cells when we talked about the red blood cells and how they change when we put them in different kinds of media. So this is the process of dehydration, and this is why water moves out of cells. So this, as you see, is a multi-step mechanism between the plasma, the interstitial fluid, and the cellular fluid. All of these always are changing and are regulating so that we maintain equal osmolarity between these three compartments. Now, let's take a look at another situation, and let's take a look at overhydration. Now we're looking at taking in too much water in the body, for example, adding only water. I want you to imagine somebody that's drinking a large volume of pure water over a very short period of time. Okay, and that's the kind of situation that we're talking about here. Notice that the osmotic pressure decreases in extracellular fluids, and water moves into cells. This is kind of the opposite case of what we had in the last slide. Let's take a look once again at the blood. And we start out by having a normal body osmolarity of 300 milliosmoles per liter. The interstitial fluid, also 300 milliosmoles per liter. And then we have a normal cell in the body, and that's also going to be 300 milliosmoles per liter. That's a starting situation. Now imagine that we take water, and we're taking water into the body, and we're adding it to the plasma first. Well, by adding the water, what that's going to do to the osmolarity, because of our equation that we looked at before, is it's going to reduce the body osmolarity or the plasma osmolarity to about, let's say, 280 milliosmoles per liter. Now, once again, what we're going to do is we're going to compare the osmolarity of the interstitial fluid to that in the plasma. You'll notice that now the plasma is hypotonic to the interstitial fluid, and that means that water is going to move this way. And when water starts to move into the interstitial fluid, it's going to dilute this. Let's say this goes down now to 290 milliosmoles per liter. Once again, we compare the intracellular fluid to the interstitial fluid, and we see that the interstitial fluid is hypotonic to the intracellular fluid, and so water is going to move this way into cells. And that's going to basically change the osmolarity inside cells. Let's say this goes to maybe, let's say, 295 milliosmoles per liter or so. Now, ultimately, all of these are going to even out just like they did before, because the fluids are going to keep shifting until we have an equal osmolarity between these three compartments. Let's say this all evens out at about maybe, I don't know, 293 or so milliosmoles per liter. You can actually figure this out exactly, depending on how much salt or water or that kind of stuff we do, but we don't need to do all that. All I want you to really understand is the major concept here. Ultimately, what's going to happen if we add water to the body is we're going to reduce the osmolarity of body fluids so that all the body fluids are now going to be hypotonic to what we normally have. Now, what's going to happen to the cells in the body? Well, once water moves into the cells of the body, what's going to happen is these cells are going to swell. Okay, so more water has moved into the cells, and that's going to cause them to swell. And this actually is connected to something that's called water intoxication, which we mentioned in the last table. And this causes confusion, hallucinations, convulsions, coma, death. It's very, very similar to alcohol intoxication. The basis of this, however, is that the cells in the brain are starting to swell. And when the neurons in the brain start to swell like this, they don't function as they normally should. And that causes this confusion, the coma, perhaps death if this goes on long enough. And there actually have been people who have taken in enough water that they've killed themselves. So this is a real thing. And some people drink so much water over such a short period of time that they can have a problem with this. And this is called water intoxication. Okay, so that finishes up this lecture on water balance. What we're going to do next is in lecture 17.2, parts A and B. We're going to be talking about electrolyte balance in lecture 17A. We're going to be talking about acid-base balance in lecture 17B.